Good afternoon, everyone. I am Professor Caterina Semendeferi. I am the Vice Chair for Undergraduate Education in the Department of Anthropology. And it is my great pleasure to welcome everyone in today's event, which is about honors thesis presentations that came out from this academic year, 2017-18, and then followed by an award ceremony. Please be mindful of any noise you may be making with your cell phones. Turn off cell phones and coming in and out the doors, because we are recording this event as every year now, and we're going to be putting it on our website for family and friends and everybody else to watch. This is the flyer. Everybody received this flyer, I suppose, for the event. And without further ado, I would like to introduce the students who are going to be coming one after the other to do their presentations. Each one of them has 15 minutes, and within the time frame they will also be taking a couple of questions from the audience. So please raise your hand and ask any question you may have. I assume one or two questions would be appropriate given the time frame we have. So welcome everybody, and I hope you enjoy today's event.
Test. Can you hear me? Okay, awesome. Well, good afternoon, everybody. My name is Cesar Casimata. I'm a biological anthropology major, minoring in biology, and I'm interested in how the environment affects our biology and its implications for health. Now, throughout my time here at UC San Diego, I've had the privilege to work with Dr. Amy Nan, molecular anthropologist, and to also take part in her research, which is focused in understanding how social experiences can become embedded in our biology and affect health throughout our life. Today, I will go over the use of hair cortisol as a biomarker of the social environment and its association with anthropometric measures of North American children, in particular those of Hispanic immigrants. My presentation will be broken up into 8 parts, beginning with what motivated my research and ending with my implications for future research. It is hypothesized that adverse conditions early in a child's life may predispose the child to chronic infection with implications for later chronic disease throughout adulthood. This is thought to occur throughout the effects a child's social environment has on cortisol production, which, if persistent, may affect their metabolic health. While it is notable that an association between socioeconomic status and cortisol exists in adults, the role in cortisol in specific pathways of adverse socioeconomic circumstances and health consequences in childhood remains unclear. Because of how vulnerable children are to adverse conditions, it is imperative that scientists understand how certain conditions in a child's environment can influence their health. This is especially true for children of minority populations, who oftentimes face strenuous conditions stemming from their socioeconomic status. Cortisol, also known as a stress hormone, it is a steroid. It's a stress hormone. Also, it is a steroid hormone produced by the hypothalamic pituitary adrenal axis, HPA axis for short, in response to stress. The HPA axis is your body's primary stress response system, and it regulates your body's adaptive response to stress. Cortisol is the end product of the HPA axis and helps cope with stress, maintain homeostasis, and put an end to stress response through negative feedback. Here's where it gets really interesting. Sorry, not yet. Here's what gets interesting. Hair cortisol has been recognized as less intrusive and preferable in assessing chronic stress over extended periods of time. This is because concentration of cortisol over an extended period of time, because concentration of cortisol in the most proximal 1 centimeter of hair approximates the average cortisol output from the HPA axis over a period of a month of growth. Because hair grows 1 centimeter per month, hair can provide information of long-term exposure to stress. The aim of this study is to gain a better understanding of the factors associated with hair cortisol in children. I analyzed the scientific literature and looked for associations between the concentration of hair cortisol in children and aspects of their social environment, such as measures related to their socioeconomic status, neighborhoods in which they live in, schools, and for children of Hispanic immigrants, measure of acculturation, which is the assimilation to a different culture, typically a more dominant one. I also looked for associations between hair cortisol and their anthropometric measures, such as body mass index and waist circumference, measures used in identifying metabolic syndrome, which is a group of consequences such as high blood pressure and abdominal obesity that may increase the risk of heart disease, stroke, and diabetes. Through this, I will present the current knowledge and gaps that exist within the scientific literature. From this research, I address the following questions. What has been published on the use of hair cortisol in children living in the US, in particular children of minority populations, such as children of Hispanic immigrants, and if there's any association between hair cortisol and their social environment, their anthropometric measures, or both. To answer these questions, I extracted data from studies only published within the US and will provide the aims, the demographics, the measures, and the results. Okay, space worked. A literature review was conducted on the use of hair cortisol in children with a focus in the most vulnerable, such as children of Hispanic immigrants. A broad search was conducted via PubMed to obtain all articles that have used hair cortisol in children and adolescents. Out of the 289 articles yielded, only 20 met inclusion criteria, and for this presentation, only 5 studies will be talked about, which were published in the US. Here are the 5 studies published in the US. The oldest study was from Palmer et al., published in 2013, and the recent studies were published last year. Looking at the aims, the studies focus on the relationship between hair cortisol and socioeconomic status, salivary cortisol, and chronic stress, and the children's social emotions. Analyzing the demographics of the going over the measures, only 2 studies assessed hair cortisol along with measures of the socioeconomic status. 2 other studies assessed it along with anthropometric measures. However, it was only based on the sex of the child. Analyzing the demographics of the 5 studies, the majority of the studies sampled were quite small. This is especially true when looking at the relatively small sample within the recent studies. Furthermore, looking into the race and ethnicity of the children from U.S. studies, we see that aside from African Americans, the proportion of minority populations is small for how diverse this country really is. Excited by the use of hair cortisol as a biomarker of the environment and its stress, I looked for whether or not there were any associations between hair cortisol, their social environment, and their anthropometric measures. In search for associations, only 2 studies provided data. The 2 studies showed associations between hair cortisol concentration and parental education, but both differed within household income. Looking at the anthropometric measures, there was no data found. That's because they focused on sex and didn't really look into the differences between sex and hair cortisol concentration. Looking into the associations found in both studies, presented in the last slide, results from the study showed that lower parental education was linearly associated with hair cortisol. However, a child's household income was not significantly associated. It was also found that infants of high socioeconomic status had significantly steeper cortisol slopes, which is a very interesting phenomenon. Presenting the current knowledge that exists within the scientific literature regarding the use of hair cortisol in children of the US, I found that only 5 papers have been published. From those papers, recent studies included small samples of minority populations. The only measure used in relation to hair cortisol was socioeconomic status and sex of the children. In regards to children of Hispanic immigrants, no measure of acculturation was found, so it's not known how acculturating to a new culture affects them. No studies took into account both a child's social environment and their anthropometric measure. This is especially true for children of minority populations. And in the two studies it discussed in our last slide, both parental education and household income are measures of a child's socioeconomic status. However, these mixed results have been consistent in the other 15 papers that I did not include for the sake of time. Furthermore, the results from infants is a very interesting phenomenon that raises the question in whether the differences between cortisol of an infant or a non-infant child has to do with either their age or at different stages of their development. Also, the researchers mentioned that looking into infants and their socioeconomic status and hair cortisol has not yet been comprehensively examined. Looking back at my questions on what has been published, I conclude that this is a fairly new study in the US. There is not yet an understanding on how the environment influences cortisol production in a child, nor what it means. This is also the case for children of Hispanic immigrants as well as other minorities that live in the US. Furthermore, there is a gap in the scientific literature that use hair cortisol in North American children, and those of minority populations. Also, more studies are warranted. Now, taking into account all the studies, the use of hair cortisol in children has the potential to be a reliable biomarker of the external effects that the social environment has on the well-being of children. However, so far there is not yet a proper understanding of how environmental stressors influence hair cortisol, nor its implications on the well-being of children, especially those of minority populations. This being the case, research that takes into account the different aspects of a child's social environment is extremely vital to understanding how exposure to early adversity influences health and how it may contribute to the diversity of adult diseases that we see common today. Further research should take an ethnographic approach and be more community-based. Further studies should be longitudinal and consist of a large, diverse sample of children from different communities from both ends of the socioeconomic scale. An ethnographic approach in a longitudinal study would allow the researchers to not only rely on hair cortisol and patient questionnaires, but also allow them to better understand the behaviors and the ideologies of the population under study, which could be of great use in order to truly understand how different lifestyles may influence health throughout life. Now, with that being said, I wish to thank Dr. Amy Nan for her mentorship and support throughout my time here at UC San Diego, and for strengthening my desire to work to combat health disparities and providing me with the resources, knowledge, and skills needed for the next chapter of my academic career. I also want to thank Professor Katerina Semendefere for her guidance and encouragement throughout this program, my honors cohort, the staff and professors from the Anthropology department who have encouraged my curiosity and opened my mind not only to the biological world, but the cultural world as well. And last but not least, UC San Diego and the Anthropology department. With this, I leave you with a quote by cultural anthropologist Ruth Benedict. Thank you. Any questions? Okay, thank you.
All right. Good afternoon, ladies and gentlemen. My name is Matthew Van Skalkvik. I'm a biological anthropologist with a strong interest in archaeology. And my presentation today is called Implications of Lithic Analysis in Understanding Socioeconomic Dynamics in the Levantine Early Bronze Age 3 and 4. My advisor for this was Professor Levy, who runs the Levantine Archaeology Lab. So here's a quick overview of these subjects that I will talk about today. I will begin by explaining what lithics are, what they can teach us, and how we can study them. I'll then give a quick crash course on the Levantine Early Bronze Age before moving on to ways that we can apply lithic analysis to learn more about the different cultural groups that made up the Levant during this time period. And finally, I'll discuss my personal research on stone tools from the site of Hirbat Hamra Ifdan in Jordan. So real quick, what are lithics? Well, a lithic is a stone tool, and stone tools are vital to our understanding of past human societies because they are our oldest form of technology and arguably date back to 3.3 million years ago. Because stone tools can be found throughout the prehistoric periods of all continents from Africa, Europe, Asia, and the Americas, and because they are present in all forms of archaeological contexts, they give us a unique window by which we can study the evolution of craft production, material culture, technology, social organization, and social complexity. So my question in this research was the following. In what ways does the lithic record of the Levant reflect the social, economic, and political climate of the early Bronze Age? Here you have the Levant. Dr. Levy works in southern Jordan on a site called Hirbat Hamra Ifdan, where the arrow is pointing, and I'll discuss more about this site later on in the presentation. So now, how do we interpret lithics? Well, lithics are essentially a form of craft production, just like basketry, textiles, or ceramics. And Catherine Costin explains that craft production embodies materialization, which is the transformation of intangible ideas and beliefs into physical objects of symbolic value. Next up, we have craft specialization. Jacob Vardy and Asa Gilead argue that specialized tool types are associated with entire bases of knowledge, and the loss of that tool type is associated with the loss of that knowledge. Additionally, Catherine Costin explains that as a society becomes more complex, so do the ideas that crafts are meant to embody. At the same time, Steve Rosen argues that lithics reflect past people's habitus or way of doing things. And finally, stone tools leave behind a strong physical trace in the archaeological record because they leave behind so much debris. So as a result, we can further study lithics through more specialized methods such as microwear analysis, mass debitage analysis, or spatial analysis, which will inform us, for example, on how lithics were organized on a site or what tasks stone tools were used for on a site. So here's a quick review of the Levantine Early Bronze Age. The first image is a comparison of the Early Bronze Age to Palestine, Egypt, and Mesopotamia. And below you have a visualization of the rise of urbanization and collapse of urbanization, which I will explain shortly. The Levantine Early Bronze Age lasted roughly from 3500 to 2000 BC and was divided into 4 sub-periods. I focused on the EBA 3 and the EBA 4 as they're the most dynamic socially, politically, and economically. The EBA 3 was the peak of social complexity in the Levant and saw the first rise of urbanization. This was embodied by larger cities and larger populations. Examples of larger cities included Yarmouth, which spanned 16 hectares, and Tel Beit Yara, which spanned 25 hectares. This period also saw the rise of trade with Egypt, as Egypt even set up a colony in the southern Levant. I'll discuss this later too. Meanwhile, the EBA 4 was the exact opposite. The EBA 4 suffered a social collapse. A possible explanation for this is the instability of the social-political system at the time. As Raphael Greenberg explains, towards the end of the EBA 3, those centers which were in charge of controlling labor and resources were simply hollow shells that were not capable of controlling labor and distributing food anymore. At the same time, trade with Egypt came to a halt. So my question here is, will we see a sign of these dynamics in the lithic record? I hope to answer this question by reviewing 3 main cultural groups of the Levant. I will go over and their toolkits. I will go over the desert region, the Mediterranean region, and the Egyptian region. So starting off with the desert. The desert was sparse and hot and sparse, so the inhabitants here could not practice agriculture. Instead, their economy was based on hunting and on trade, which is reflected in the stone tools that this area produced. Firstly, we have arrowheads, which were used in hunting, and secondly, they also made microlith drills used to make beads, which were then used in trade. More importantly, this area is most famous for the tabular scraper, a large fan-shaped stone tool used for cutting and butchering, not scraping. And these stone tools were exclusively produced in the desert and then traded with other areas in the Levant. In the meantime, the Mediterranean region was coastal and temperate, and this allowed for the development of agriculture. This is also reflected in the higher level of specialization of lithics in the region. So firstly, you had a set of ad hoc tools, or the Swiss Army knife of the Levantine farmer. These tools were simple, made on the go, and were used for a variety of daily chores such as cuddling, piercing, or whittling. They also produced what is known as the caninian blade. So these tools were used to cut and reap grasses, and we know this because of the characteristic gloss or luster left on their edges from the smearing of plant silicas. Of note, although the tabular scraper was often traded into this region, caninian blades were never traded into the desert because they had no use for that tool over there. Finally, the Egyptian region had much more specialized tools. This is simply because they were the result of a much more specialized chain of production, and also because Egyptian lithic knappers went through a much longer period of apprenticeship. All this is related to the fact that Egypt at this time was much more hierarchical than the Levant. Also, the tools here can help us answer an important question. Was the Egyptian colony in the southern Levant peaceful or militaristic? Well, it was peaceful for many reasons. Firstly, Egyptian lithic assemblages have only revealed blade-like tools for agriculture and no arrowheads which would have been used in warfare. Secondly, since tabular scrapers and caninian blades are found in Egyptian lithic assemblages, we can safely assume that the population here was here to practice trade with the local population and not to go to war. So overall, how did these three lithic toolkits reveal of the economic, social, and political climate of the Levant? Well, economically, the lithic styles reflect the environment that produced them. Politically, the urban breakdown at the end of the EBA-4 actually put an end to both tabular scraper and caninian blade production. And socially, we know that the Egyptian colony in the southern Levant was peaceful and not hostile based on the character of the lithics they left behind. On the left, you have an example of the tabular scraper, in the middle, caninian blades, and on the right, you have an example of what the Egyptian lithic toolkit looked like. I would now like to focus on my personal work and research on stone tools from the site of Herbat Hamar Ifdan, located right below the Dead Sea. So here's a very quick history and overview of the site. So located in southern Jordan, the site was first visited in the 1930s by Frank and Gleck, but was not excavated until the 1999 and 2000 field season by my advisor Thomas Levy, Russell Adams, and Mohammed Najjar. And KHI is most well known because it is the largest metallurgical factory in the ancient Near East, with about 3,782 artifacts uncovered. KHI is also particularly interesting because it can be divided into 4 strata or layers, with each stratum belonging to a different phase of occupation. Stratum 2 and 3 belong to EBA 4 and EBA 3 occupation. So as a result, KHI provides a cool window by which we can study both periods of time individually and how the site was affected by both social complexity and by urban collapse. Here my question was, does the lithic record of KHI agree with our knowledge of the early Bronze Age? And I tried to answer the question by doing a debitage analysis and then a spatial analysis of the tools of this site. So what is a debitage analysis? Well, it involves looking at each individual stone tool from a given assemblage or set of stone tools and then assigning it to a type based on surface features. For my analysis, I looked at the 99 collection of debitage from KHI, which took me through about 7,500 pieces. So here you have a summary of the tool types from this collection. And using this table, I was able to make some calculations, which helped me come up with a preliminary idea of how stone tool production was organized at the site. So I found that only 14% of the debitage was found in area H of the site. 2/3 of the cores were found in area Y. 43% and 50% of the blades were found in area C, and finally 43% of the flakes were found in area H. So based on this, I came up with a hypothesized chain of production going from area H to area Y to area C. Additionally, using the site's master loci list, I calculated how much stone tools were found in each stratum of the site. I found that 0.15% of the lithics were found in stratum 4, 46% were found in stratum 3, or the early Bronze Age 3 occupation, 29% were found in stratum 2, or the early Bronze Age 4 occupation, and finally 25% were found in stratum 1. So what does this say? Well, as expected, there is a correlation between intensity of lithic production and the time period, which supports the well-known idea that the EBA 3, or the peak of the early Bronze Age, was indeed where most of the social and economic activity was happening. Now moving on to my spatial analysis. So I created these maps using an app called GIS, which allowed me to point out where lithics were concentrated on the site. So there are 2 sets of maps, one for stratum 3 and one for stratum 2. And for each stratum, I created a map of chunks, cores, flakes, and blades. So what do these tell us? Well, in comparison to stratum 3, the range and volume of stone tools is much greater in stratum 3. In comparison to stratum 2, the range and volume of stone tools is much greater in stratum 3, which is easily explained by the fact that that stratum 3 was the main occupation phase of the site, which was associated with the early Bronze Age 3. Chunks are distributed relatively evenly throughout the stratum, so it is safe to assume that this is due to the fact that chunks are the most common type of waste produced during the knapping process. Meanwhile, cores are concentrated in the center of the site in an area known as structure 54. Blades are concentrated in the northwest corner of KHI around structures 36, 37, 42, and 46. And one hypothesis could be that finished blades were stored in this area of the site after being produced in structure 54. Now moving on to stratum 2. The greater scarcity of stone tools found in stratum 2 is an indicator of the reduction in socioeconomic activity that happened as a result of the urban collapse of the early Bronze Age 4. Unlike stratum 3, where all types of debitage are found in greater abundance in certain areas of the site, all debitage is scattered about evenly throughout this stratum. This could mean that although stone tool production was still being practiced at KHI during the EBA4, it was no longer a specialized activity, and it was now practiced in a more ad hoc fashion. So all in all, the lithic density maps of stratum 2 and 3 of KHI lend support to our current knowledge of the Levantine Early Bronze Age, where the Early Bronze Age 3 was indeed the most complex in terms of social, economic, and political activity. So to conclude, I'd like to rephrase my first question. In what ways does the lithic record of the Levant reflect the social, economic, and political climate of the Early Bronze Age? Well, lithic toolkits and tool types reflect the environment in which they are made. For example, you have desert versus agrarian societies. Secondly, lithic production was dependent on the social and political climate of the period. For example, the EBA4 urban breakdown put an end to both tabular, scraper, and Canadian blade production, and the Egyptian colony in the southern Levant was peaceful and not hostile, based on the character of the lithics they left behind. Thirdly, lithic specialization is also dependent on degree of social complexity. For example, we see greater level of variation and specialization in the Egyptian lithic toolkit compared to the desert and Mediterranean regions. And finally, spatial distribution and intensity of lithic production at KHI was much greater during the EBA3 than it was during the EBA4, as expected. Thanks for listening. You guys rock. That's a pun. Any questions? All right. Oh, yes.
So I was wondering, when you were going over the different layers of strata, did you find all that information via research through library research, or through actually going through it?
Oh no, I went through boxes of stone tools and looked at them one by one. Yeah, and then I went through their records, and I matched them with which stratum they belong to. Yeah.
Tell me how many you went through.
7,500. Yes.
Would you expect the transition from stone tools to the really fancy metal tools to be super gradual, with only the dominant?
Yes, it was actually very gradual. Yeah, because KHI was actually the biggest metal factory, so that was very cool to compare the metal stuff to the lithic stuff, and it was actually very gradual, not once.
But you have such a time span.
Yeah, it's so large. Yeah. Do you have more time for questions? Okay, I'm sorry, I can't take your question.
Well, it's one more.
Oh, one more? Okay, yes.
Well, I first wanted to mention that I'm sorry for having to listen to your...
oh, nice.
And I was curious, I think you mentioned that at the end of EBA 3, there was a minor reduction in specialized tool use. Is this for any particular reason?
Yes, what happened is a lot of the population left big cities, and as a result, they couldn't produce the stone tools anymore because that knowledge was lost. Because of the urban breakdowns, they all left the cities, and as a result, they didn't produce stone tools anymore. All right.
Okay, so good afternoon. My name is Joshua Beneventi. I'm a graduating senior majoring in the field of sociocultural anthropology, and today I'm going to be presenting to you the findings of my undergraduate research thesis titled Dissolution and Transcendence, the Relationship Between Music and Altered States of Consciousness. Before I begin, I would like to express my deepest gratitude to my mentor, Steve Parrish, to whom I give my deepest thanks for his unprecedented gift of time and guidance throughout this whole process. I'd also like to thank Katerina Semendeferi and the honors cohort. We made it. So before I begin, I would like to start with a brief overview of our journey together spanning the next 12 and a half minutes. First, I will introduce the topic and present my primary research questions. We will then delve into the ethnographic material that I have drawn upon and juxtapose that with findings of prominent researchers in this particular field. We will then, from that analysis, I will draw my primary conclusions, which will then take us to the discussion of deeper concepts, reaching to the heart of what I believe is the societal structure. So without further ado, let us begin. Over the course of my studies and travels, I have observed a recurring cultural phenomenon that totally grasped my attention. That being the widespread institutionalization of altered states of consciousness, or more easily put, trance. This is a phenomenon that can be found at the heart of societies the world over. In a cross-cultural study of 488 societies, it was reported that around 90% incorporate consciousness-altering rituals in their religious systems. Ritualized methods of induction to the altered state vary widely, for instance, through dance, psychoactive substance consumption, fasting, etc. However, there is one technology which seems to be central in this ritual process across the board. That being music. Music has shown to be the most cross-culturally prevalent factor in altering states of consciousness and acts as a key ingredient in the successful induction process. Having observed this prominent connection between music and the altered state at the core of diverse religious institutions, I posed the following questions. Primarily, what are the agentive factors inherent to music which cause it to assume such a prominent role in altering consciousness? And in succession, what is the overarching significance of such practices, both to the society and the individual upon whom the phenomenon is being embodied? So I'd like to talk a little bit about what it is that we're talking about when we say altered states of consciousness or trance. The word trance comes from the Latin transire, which means to cross over or transition. And this can be defined as a set of altered states of mind that bear only family resemblance to one another. So it's essentially a constellation of related symptoms. Gilbert Rouget was one of the foremost researchers in this field, and he loosely categorized different manifestations of trance into ecstasy and trance. And ecstasy is associated with sensorial deprivation, things like silence, fasting, darkness, immobility, and recollection of the experience, whereas trance is associated with sensory overstimulation and has to do with situations involving music, noise, agitation, and oftentimes amnesia, which brings me to the particular form of trance called possession trance, which is going to be what we're talking about today. And this can be defined as a situation in which a person is changed in some way through the presence in him or on him or her of a spirit entity or power other than his own personality, soul, self, or the like. The main body of ethnographic literature that I sourced in my research focused upon the West African diaspora religious cults of the New World, in particular one Brazilian variant called candomblé. Though this thesis was mainly a literature review, my own personal experiences in Brazil did play a significant role in ethnographic descriptions. Candomblé is a syncretic religion brought to the Americas during the forced migration of various West African populations through the Atlantic slave trade. There are numerous denominations within the cult. However, the dominating cultural influence, which has left its mark in modern times, came from the Yoruba people, originating in what is now known as northwestern Nigeria and Benin. The candomblé is technically a monotheistic faith. However, it is commonly considered poly or pantheistic due to its cultivation of numerous divinities called orishas. And here we just have some quintessential images of the candomblé in Salvador, Bahia, Brazil. So essentially, all I want to leave you with about the candomblé is this word orisha. So these are the divinities. They're ancestral. They're associated with ancestral locations in Western Africa, as well as forces of nature. So they're kind of elemental. They're appeased through sacrifice and offerings, and ultimately drumming ceremonies. The goal of all religious activity in the candomblé is to manifest the orisha in the predetermined mediums during these large religious ceremonies. right, now let me see if I have a really quick clip here for you, which... All right, so that's just a little clip of a ceremony at the moment when the medium is being possessed by the saint. So Candomblé, essentially our structure is this, that it's a syncretic religion centered around spirit possession by the Orisha, which is achieved through this process of ritual sacrifice offerings, etc., culminating in these ceremonies. The music of Candomblé, let me play this. The music of Candomblé is a religion in which music is so ubiquitous that the same term encompasses what we recognize as music and religion. The drums are considered divine and actually treated as orishas themselves. Drumming accompanies all major ceremonies, and during the drumming, the players of the drums as well as the officiants of the ceremony make conscious modifications in the rhythms in order to induce the altered state in the mediums who are dancing. And then for individuals who are chosen to be mediums, the cloistered initiates, they are exposed repeatedly over extended periods of time to these drumming rhythms, which is essentially a sort of hypnotic entrainment where they receive information about the cult and their saints, etc., etc. So this is the ethnographic example that I've used in order to discuss deeper concepts of the sociocultural structure, which I believe it saliently exhibits. So first, looking at it from the sociocultural perspective, the function of music is to create a special emotional atmosphere which stimulates processes of identification within social groups. It is trance-inducing, accompanying, and/or guiding. Music is considered a technology which enables the loosening of perceptual boundaries, collapses boundary between subject and object. And then I'm not a biological anthropologist, or neither do I have any background in neuroscience, but just touching upon this aspect of it to provide some substantiation. Music, it has been shown to interact directly with structures in the brain associated with emotion, particularly the limbic system. We know that music is associated with dopamine production, and I found this very interesting from a ritualistic perspective. Phasic increases in dopamine release happen when meaningful objects are in the focus of attention. The higher the personal meaning and valence of the objects in focus, the more dopamine is released in the brain. And finally, music functions as a catalyst of strong emotions that may lead to trancing. So we know that there are these two clear aspects to it, the objective biological effects and then as well the socioculturally conditioned effects. So the altered state is brought about primarily through social conditioning, the habituation to certain musical stimuli, which is inextricably connected to the physiological effects that the sound vibrations have upon the organism, both directly and through the conditioning process. And this brings me to my preliminary conclusion, to my primary question. What are the agentive factors inherent to music which cause it to assume such a prominent role in altering consciousness? Like I just said, the effects of sound vibration upon the organism and the social cultural conditioning, which are inextricably connected. And at the core there, the third part of this process is the aspect of emotionality. Music engages psycho-emotional group bonding processes, and it functions as a catalyst of strong emotions that may lead to trance. What causes a music to appear so prominently across the board in diverse cultural settings is precisely its ability to transcend the boundaries and regulatory functions of the centers in our mind associated with language, logic, and reason, and taps into deeper centers of emotionality. And I like this quote by Jankowski: "Immediate connections with others can be made through music. It emphasizes what connects us as humans rather than what sets us apart as social factors." And with that, I would like to address my second question, which is a little more conceptual, and that is the overarching significance of such practices to both the society and the individual upon whom the phenomenon is being embodied. And that it is essentially has to do with social cohesion. And we know that ritual is a periodic restatement of the terms in which men of a particular culture must interact if there's to be any kind of coherent social life. So we know that social cohesion is absolutely necessary in the proper functioning of society. But to take it a little deeper, I looked into the work of Victor Turner and Emile Durkheim. And Victor Turner's concept of communitas, he proposes that the communitas could develop through the liminal phase of ritual and liminoid phenomena such as trance. And Durkheim recognized that ritual is powerful. It has effects on people and does not simply make them feel stronger, it makes them stronger. Collective effervescence is defined as communal participation in singularly focused activity or thought, which allows for emotionally charged transcendent experiences. So this aspect, the anti-structure is what Victor Turner calls it, of the liminoid phenomena. It is a place where society's values, norms, and deep knowledge of itself are reaffirmed and sometimes created. And it is in these effervescent social milieu, and indeed from that very effervescence, that the religious idea seems to have been born. So as far as this last question is concerned, I cannot give a solid answer, but I think it is quite clear that there exists a profound relationship between the ideas of structure and anti-structure, both entirely necessary for the existence of the other. And in this symbiotic dance of structures, we find something very unique which brings us to the heart of culture and the heart of humanity. And I think this quote here by Durkheim, where the religious idea seems to have been born, is very, very significant in that. And I'd just like to leave you with this final quote. Music and consciousness are things we do. Achieving consciousness, from the Latin con, which means with, and cire, which means to know, is the central activity of human knowledge. At the heart of this word is a concept of mutuality, knowing with others. Our consciousness is a mutual activity. It is performed. Thank you. Are there any questions? Yes?
Thanks for a fascinating talk. I have a hypothetical question. So
Sure.
Che Guevara, who cannot be accused of not having cared about conscience and society, suffered from amnesia.
Amnesia?
No, amnesia. He was incapable of singing a tune or dancing to a beat. And
Interesting.
He was very familiar with Yoruba culture and the Orisha in Cuba. Do you think he could have fallen into a trance?
Yes. Yeah, most definitely. I think that the induction method, and as far as my research is concerned, I mean, you can, nearly anything could be considered an induction method, just the intention. And that's actually very inherent to the process. It's necessary that the individual be participating in the culture and that all of this has some value to them and that they be willing to fall into the trance. So I think it's totally possible. Thank you. Anybody else? All right, thank you very much.
Good afternoon. Thank you for being here. My name is Barbara Collingford. I'm an anthropology major with concentration in biology. My thesis advisor is Dr. Pascal Gagnon, and I will present my research on assistive reproductive therapies and human reproduction. During this presentation, I will cover the following: an introduction of the topic, the questions that I expect to answer with this presentation, a brief history of assistive reproductive therapies, as well as the impact that they can have on epigenetic mechanisms. To end with, I talk about potential effects of cryopreservation in culture media, and I discuss some of the future work that could benefit this topic to end with my conclusions. Over the last 4 decades, ARTs, or assistive reproductive therapies, have grown in popularity and have gained people's trust by offering alternative ways to treat infertility. Nevertheless, they are imperfect and must have an impact on reproductive health. I focus on 3 aspects of reproductive health that might be influenced by ARTs: growth, development, and genetic and epigenetic mechanisms. The research questions that I hope to answer this evening are the following: how can assistive reproductive therapists impact human reproduction? and more specifically, which genetic and or epigenetic processes are altered during assistive reproductive therapies, and what is the meaning of this. The methods used for this research were a literary review of biomedical articles and books, and I focused my research on the consequences of using assistive reproductive therapies, human reproduction, effects on the genome being genetic or epigenetic. Mammalian fertilization happens when two haploid genes, an egg from a female and a sperm from a male, interact and fuse to create a diploid zygote, which in the majority of mammals develops in the utero during the gestational period and coexists via the placenta, which is made by the baby, not by the mom, in intimate contact with the mother and her vasculature. However, a number of physiological and immunological processes are required in the female reproductive tract in order to allow both gametes to meet, recognize, and fuse with each other. Humans, compared to other mammals, are relatively infertile. The rate of monthly fecundity in humans is of 20 to 25%, and about 10% of couples in an industrialized society will have some issues with subfertility. At the same time, there's been an increase in individuals who delay their reproduction due to different reasons, such as pursuing a higher education or getting a job more demanding. The quality of the gametes produced by both males and females diminishes as we age, so age is an important factor in fertility. Both male and female are negatively affected by obesity, as well as other lifestyle effects. Some of the consequences from obesity can cause ovarian dysfunction and anovulation, which refers to the lack of oocyte release during a menstruation cycle. This affects fertility and can cause delays in the time to pregnancy, as well as poor embryo development, and ultimately can lead to pregnancy loss or an increase in risk of birth defects. At the same time, a higher body mass index in male can cause altered sperm production and can compromise chromatin integrity. Lastly, couples with obesity problems might have a lower success rate even after going through assistive reproductive therapies. Fertility plays a primordial role in the development of all species. Nevertheless, it is not guaranteed for every individual. Over the last century, scientists have developed a number of technologies which are aimed to improve reproductive challenges in both males and females. These technologies are referred to as assistive reproductive therapies, and over the past decades, their rapid development has made it possible for a lot of individuals to conceive offspring. Although these therapies have been pursued extensively to mimic an in utero pregnancy, there are still several aspects of these technologies that warrant further investigation due to their potential impact on the development and growth of individuals that were conceived through them. The scientific debate often notes that scientific development comes with a series of consequences. So close attention should be paid to not only children conceived through ARTs, but also the mothers who carried the babies. I wanted to include a timeline that presents some of the landmarks of assistive reproductive therapies to show how they evolved and also to make emphasis on the fact that these therapies are really recent. For the longest time, it's been known how to collect male sperm. Nevertheless, collecting oocytes is a lot harder. In 1925, pituitary hormones were discovered, and later in 1930, the human chorionic gonadotropin, which is a hormone that induces ovulation, was used in order to collect more oocytes. Ovulation induction, which is an assistive reproductive therapy, refers to the stimulation of ovulation in females through some type of medication. It's a very important and common therapy that should be carefully followed, since the mother's health could be compromised during and after the process. In 1978, we have Louise Brown, who was the first baby born through IVF in the UK. IVF, or in vitro fertilization in brief, consists in the collection of sperm and harvesting of eggs after hormonally inducing them, followed by combining this in a culture medium in order for fertilization to occur in the outside of the female reproductive tract. In 1982, we have the first successful intrauterine transplant of an embryo. And in 1986, we see more ways of retrieving oocytes and the first donated oocyte pregnancy or surrogate mother case. In 1992, we had the first intracytoplasmic sperm injection baby. And in the 21st century, We have the beginning of pre-implantation diagnostics, embryo, and genome editing. After fertilization, some cells of the newly formed embryo migrate to the germinal ridge and will eventually become the germ cells, sperm or oocytes. Due to the phenomenon of genomic imprinting, the DNA of maternal and paternal genomes are differentially marked by chemical modifications, and they must be properly reprogrammed every time they pass through the germline. Major epigenetic reprogramming occurs twice in our lives: when our germ cells are developing and during the young embryo prior to the implantation in the case of assisted reproductive therapies. These two stages are a part of both in vivo and in vitro fertilization, and there could be interferences due to the assisted reproductive therapies with the proper establishment and maintenance of genomic imprints. Intracytoplasmic sperm injection, or ICSI, is a form of assisted reproductive therapy in which a single sperm cell is injected into the cytoplasm of the egg. ICSI does not require functional sperm. Therefore, not a lot of spermatozoa are needed. As a result of this, the spermatozoa do not pass through the filter that the female reproductive tract faces, as well as the vestment and the zona pellucida. So there can be a result of fecundation of the egg with defective sperm. Imprinted genes play a very important role in embryonic growth, behavioral development, and are involved in carcinogenesis due to their function as tumor suppressor genes. Evolution of genetic imprinting is likely a result of a conflict between fathers and mothers with regard to parental investment in mammals. The timing of imprinting of genes is crucial. Imprinting alterations may cause growth disturbances because they play a really important key role in development. An example of what could happen when there are problems with epigenetic reprogramming, with genetic imprinting is Beckwith-Wiedemann syndrome, which causes patients to have abnormalities at chromosome 11p15, which is associated with organ overgrowth and abdominal wall defects, as well as increased risk of embryonal tumors. DNA methylation likely represents an ancient defense mechanism by which an organism can defend its own genome against selfish transposable elements that could spread and introduce deleterious mutations upon inserting their new additional copies in new places across the genome. In mammals, DNA methylation has evolved as a way to mediate the conflict of interest between males and females in the offspring. There is a study on superovulation inducing alterations in the epigenome of zygotes and results in differences in gene expression at the blastocyst stage. As we can see in this image, normal physiological signals equal a healthy embryo. Nevertheless, altered or stressful signals equal an altered metabolic state. Now I'm going to talk about cryopreservation in culture media. Cryopreservation is the practice in which living cells or tissues are preserved at very low temperatures in order to prevent physical or chemical degradation or enzymatic activity that might be deleterious for the cell or tissue. Critics of cryopreservation put the blame on the process of preservation itself, especially on the damage that freezing procedures can have in the cell, such as rupture or changes in composition. A study done in 40 semen samples from different men concluded that preparation of semen after cryopreservation resulted in decreased sperm motility. Culture media. Embryos formed through IVF show altered levels of mRNA due to the presence or absence of serum in the culture medium. Serum is extremely important in culture media because it provides the cells with amino acids, proteins, vitamins, lipids, and other growth factors that otherwise the female body would normally provide. In an article by Chronopoulou, it is mentioned that the amount of glucose used in different culture media affects the cleavage rate. In vitro culture has been associated with lowered birth weight, and it's believed that this occurs as a result of epigenetic alterations being accumulated while the embryo is developing in the culture medium. The potential for future work is greatly impacted by the novelty of the topic. ARTs are a massive ongoing experiment, which means that there is room for improvement, as well as the need for more research on the impacts that they could have in human reproduction. More research and follow-ups of children conceived through assistive reproductive therapies in order to have a better understanding are needed and assess the impact that the therapies may have. An analysis of the ethical dilemmas that go hand in hand with ARTs and reproduction is also needed. Cultural studies would be important to understand the different points of view of assistive reproductive therapies across the globe. So back to my question, to how can assistive reproductive therapies impact human reproduction? We can conclude by saying that some of the alterations in genes and epigenetic processes have been proven to have deleterious consequences in the development of the embryo. The novelty of the topic calls out for more collection of data regarding the presence of epigenetic mechanisms in reproduction, especially of DNA methylation in individuals that were conceived through ARTs. It remains to be seen if the over 5 million people conceived through assistive reproductive therapies will age normally. Making reproduction possible for individuals who could not otherwise reproduce could also mean propagating loss of function mutations, which result in gene products having less or no function. ARTs influence and alter the natural mechanisms of reproduction, specifically the genetic processes. Analyzing and understanding them, the relationship between the natural and artificial processes of reproduction is of great importance in order to minimize potentially adverse effects of assistive reproductive therapies, not only in the embryo, but also in pregnant women. I would like to thank my advisor, Pascal, Dr. Katerina Semendeferi, for always pushing us to do our best, my honors thesis cohort, and the staff and the faculty of the Anthropology department. Thank you.
Take a question or no?
Does anyone have a question or two? I know it.
I'm really interested in this question. I have a friend who studied epigenetic changes in imprinted genes with ART, and she didn't find any loss of imprinting, I think in the placenta. I'm wondering, since then, like 5 years ago, did you find studies that show specific changes in imprinted genes or other genes related to ART?
Most of the articles, because of that I used to do this, were written after 2011. That was my limit. So I don't know. This might be like.
Did you find examples in recent years?
Yes. Well, there's like the Beck-Wiedemann syndrome.
Related to ARTs?
Related to ARTs, yes. Related to the changes, especially of the cryopreservation in the culture media. So it's related to that. There are not a lot of articles on this, though. I did read the paper that you're talking about. But I hope that kind of clarifies it. Does anyone else have a question?
Thank you.
Okay. Hi, everyone. My name is Amy Hostetler. Today I'll be presenting on the use of chimpanzees and bonobos as models for human evolution. This presentation is based off of my senior thesis, which was a literature review I performed under Professor Shirley Strum as my faculty advisor. In the following presentation, I'm going to go over the following: the importance of this research in understanding human evolution, questions I will address throughout the presentation, background on modeling chimpanzees, bonobos, humans, and hominids, all of which I'll explain later. The use of hominid modeling, especially involving cognition and culture and aggression and altruism. And the conclusion, which I will hopefully answer the questions that I put forward. So if you think about how many times you've seen headlines on Neanderthals or other extinct human ancestors, you may be able to see how often we compare ourselves. We base a lot of our assumptions based off of our evolutionary past, and including how we talk about what it means to be human and our identity. Much of what we think about our ancestors comes from the behaviors we study about closely related primates, which is essentially what referential modeling refers to. We can recognize then that referential modeling has a huge impact beyond academics and into the public sphere. Moving on, the main questions I address are as follows. How are referential models used to make interpretations on hominin behavior? And next, what are some differences between hominins and reference that complicate the use of referential models? And here I use the term reference to refer to like bonobos and chimpanzees that are used in referential models. My third question is, what should we change moving forward? And finally, and what I believe is the most important question is, how does this change the way we think about humans? To start off with, I'd like to go over the different types of modeling. Referential modeling is more broadly defined as studying aspects about real subjects by using reference in their place, usually because it is unethical or unfeasible to be testing on the intended subject. We can see referential modeling used in many real-world scenarios, such as the use of lab animals to test human diseases, or the use of extant or modern species to recreate paleological remains. Conceptual modeling, on the other hand, refers to abstract rules and guidelines that predict how the world around us works. Familiar forms of conceptual modeling include the laws of gravity and natural selection theory. As I previously alluded to, there are problems with using both models individually to model hominin behavior. There is no conceptual model for human behavior, as there is no single law that dictates how humans specifically evolved. Referential modeling, on the other hand, suffers as well due to variation between humans, chimpanzees, and bonobos that I will make more clear as we go on. To start off with, there are several superficial traits in chimpanzees, bonobos, and hominins that we use to identify them. Chimpanzees are identifiable by having larger size compared to bonobos and tanner undertones in their skin. Bonobos, on the other hand, are smaller and leaner, which is why you may hear them called gracile or pygmy chimpanzees, and additionally have more hair tufted around their ears and bright pink lips. Like humans and unlike chimpanzees, they are neotenous, which means that they have a lot of traits that from adolescence that they retain into adulthood. Hominins, such as Lucy in this image, who is a member of Australopithecus afarensis, encompasses a larger variety of genuses and species that make up our ancestors after splitting off of the last common ancestor with chimpanzees and bonobos. But they are more easily identifiable for their hip and leg structure that denote them as bipedal. The following chart goes into more similarities and differences between the species. An interesting observation is that there are more similarities between chimpanzees and bonobos than both species have with humans, which is most likely due to their divergence occurring much sooner than our divergence with their common ancestor. There are 4 primary aspects of hominin behavior that I chose to focus on for my thesis. The first is sex-biased behavior, which I define here as behavior that is primarily done by one sex as opposed to another. One major hypothesis that came out of sex-biased behavior modeling is the gathering hypothesis, in which observations of female chimpanzees using tools more often than males led to the suggestion that foraging by women in human evolution led to a steadier source of nutrition that influenced our current behavior. Next is tool use behavior, particularly regarding the difference in tool use between bonobos and chimpanzees. Bonobos are not as prolific tool users in foraging contexts as chimpanzees, but arguably use many tools in social contexts. Either way, there is a lack of data on the specific paleoecology of chimpanzees and bonobo habitats, but knowing more about their different environments may lead to a better understanding about what led to their different behaviors. As I mentioned before, I also researched cognition and aggression, both of which I intend to discuss in detail since I have found that these types of research are both popular subjects to study and have more tangible implications on how we view ourselves. The sociality of bonobos and chimpanzees provide important insights into the cognition of both primates. Many social animals work off of behavioral rules, which are predictors about what an individual is going to do in a situation based off of knowledge from similar scenarios, which in turn reinforces the rule in a positive feedback loop. When faced with novel situations, or if an observed individual is unsuccessful, there is nothing to really give the observer an idea as to what the goal of the behavior really was. Chimpanzees and bonobos, as well as humans, display more complex goal-oriented knowledge. However, chimpanzees do not seem to have the same sense of false belief like we do. In trials where food is moved between 2 occluders in absence of when a dominant chimpanzee is absent, as shown in the figure, the subordinate chimpanzee does not seem to understand that the dominant will presume that the food is in the first occluder that he first witnessed. And this is just one example of fundamental cognitive differences between chimps and humans that have implications on our evolutionary history. Another example involves culture. I acknowledge that there are many definitions of culture that range from being centered around humans to involving more generalized forms of cultural transmission, but for the sake of this presentation, I prefer the definition of culture as the way we do things. The difference between populations of a species or each group's traditions can be indicative of the ways we do things, although I found that there are less examples in bonobos due to their limited range and low numbers, and but there are differences in tool use between humans and chimpanzees that we can examine. Culture can be delineated as being either multi-tradition, where a group employs multiple traditions as it sounds, or an expansion of that into cumulative culture, which is a multi-tradition culture built upon many generations. Human culture may be more indicative of cumulative culture since seen in the transmission and building upon technologies like refined stoneflake tools rather than the multi-tradition culture of chimpanzees who don't seem to refine tools to the same degree. It is worth noting, however, that chimpanzees do show cognitively demanding behaviors like using the same material or the same tool for multiple purposes, so it is important not to diminish their mental capabilities. In this video, a female chimpanzee uses a stick to go ant dipping, where she collects ants by forcing a stick through a trail or a hill of ants. In this case, she pulls the stick up and then runs her hand over it, over the length of the stick to collect them on her hand for consumption. In other populations, however, chimps show differences in ant dipping, such as eating ants directly off of the stick or putting the length of the stick in their mouth for consumption. Another comparable aspect between humans and chimpanzees is how we express aggression. Lethal aggression is the act of killing a conspecific or another member of the same species with the intent to kill. This occurs throughout the animal kingdom, but it's particularly prevalent in chimpanzees, with popular publications such as Demonic Males, the book, suggesting that chimpanzees have a deliberate search for victims to kill. On the other hand, bonobos do not appear to display lethal aggression in the same manner, with only one instance of suspected conspecific killing among 4 bonobo communities within 5 decades of observation. Additionally, analyses on chimpanzee aggression and hunter-gatherer warfare show similar patterns of warfare when we consider factors such as periods of peace and violence, avoidance of conflict, and other aspects of conspecific violence. However, we generally treat aggression much differently than chimpanzees, since we tend to consider heightened aggression a pathological issue. There is also the issue of bias when we talk about human aggression, since research on aggression within chimpanzees and the application of research to hominins came about right after World War II, when there was influence from fascination with and sexist attitudes about warfare and conflict. Unlike chimpanzees in warfare, bonobos are more closely associated with peacefulness and altruism due to their prosocial behaviors toward other members of the same species. For instance, as displayed in this figure, bonobos will help other unfamiliar bonobos obtain food and are prone to yawning when viewing an unfamiliar individual yawn. Chimpanzees tend to only yawn when viewing familiar individuals yawn, and will only assist another individual if they are familiar with the other one and if they are asked to help using begging gestures. As we are all familiar with, humans have similar capacities for altruism like bonobos, even in situations where chimpanzees may not act accordingly altruistic. Factors such as our culture may have an influence on our complex, aggressive, and altruistic tendencies, and potentially could have also affected our hominin ancestors. Previously, we have used referential models as one-to-one analogs of hominin behavior. However, as I have demonstrated, there are nuanced differences between hominins, humans, and our closest relatives that bring into question whether these direct comparisons are enlightening or, conversely, limiting to our understanding of hominin behavior. This conclusion affirms current trends away from using single-species referential models. We cannot view our own characteristics as somehow occurring outside of natural selection, but we are unique creatures. We're different from chimpanzees, bonobos, and every other species on the planet. And rather than making one-dimensional direct comparisons, we should recognize our complexities and look instead to better understanding how evolution shapes all of our histories. I'd like to thank Professor Shirley Strom for taking me on as her thesis student and being accessible throughout the process, Professor Katerina Simendeferi for organizing the program and supporting us, my fellow honors cohorts for valuable feedback and emotional support, and finally my friends, family, and loved ones for putting up with how much I talk about primates. So, if there are any questions about what I present, I'd be happy to answer them now.
So, going back to the altruism and I guess aggression with chimpanzees and bonobos.
Bonobos, yeah.
So in a previous class, I had learned that chimpanzee aggression isn't as high as we may think it is. Mm-hmm. Do you think that is kind of something that you would use for your argument that they're more unique than we think, or is that kind of, would you see that as kind of threatening? Yeah, so what I believe is that essentially chimpanzee aggression is definitely overstated to some extent, but they also have very unique structures in regards to aggression that are very different than what we see with bonobos and humans, and which also have their own differences. So it's not so much that chimpanzees are more aggressive, but that we just have a lot of differences that make it difficult to make direct comparisons. Any other questions? Okay.
Good afternoon, everyone. My name is Emily Rebleza, and my advisor is Dr. Hannah Garth. My thesis is titled Shaping Our Understanding of Food Justice Through Traditional Native Food Systems. To start off, I will first talk about the questions that had led me to research and aim a thesis. I will then introduce what food sovereignty is as opposed to food security and what scholars have said about the two concepts. After my methods and a quick introduction to the context of many Native communities in the U.S., I will give an overview of several structural factors leading up to the food sovereignty movement today and how communities have called for resistance through indigenous, value, and culture-centered programs. I will then add to the existing literature of how anthropology can add to food justice. I first asked the question, how has the indigenous traditional food sovereignty movement shaped how natives have come to view their cultural identity? I do so by first gathering evidence of native resistance and resilience that has made an impact on the food sovereignty movement through the existing ethnographic accounts and primary sources. In answering this, I then ask, how can this indigenous cultural identity then shape and influence scholars and policymakers' understanding of how food sovereignty can better address food injustices and that these communities face through anthropology? So first of all, what is food security and food sovereignty? There are many definitions to these two concepts, but food security is defined as all people at all times having physical, social, and economic access to sufficient, safe, and nutritious foods that meet their dietary needs and food preferences for an active and healthy life. Food sovereignty, on the other hand, is defined as a community's power to democratically manage productive food system resources such as land, water, and seeds, and to engage in trade on their own terms rather than being subjected to speculation through international commodity markets. Several authors have stated how food sovereignty differs from food security on another level, such as that food sovereignty is the best solution for food insecurity. Food sovereignty is a much deeper concept because it proposes not just guaranteed access to food, but democratic control over the food system from production to processing to distribution, marketing, and consumption. And scholars view food sovereignty efforts as being forms of resistance against the effects of settler colonialism, but also a resurgence of indigenous forms of authority and autonomy. My methods used was a literature review, which included not just works by anthropologists who have worked alongside Native community members in their fieldwork, which in a way primarily serves my paper as ethnographic accounts, as well as scholars from other social science fields, but also works by Native indigenous authors, scholars, activists, and members of community of communities who give vital voices and insight to their struggle of keeping their traditions and people alive, and have written about the concepts of sovereignty, structural issues, and of food justice itself. To give a little bit of context first, the Native American population in the U.S. has some of the highest rates of diabetes of any group of the U.S. and has one of the highest rates of obesity according to the Center for Disease Control and Prevention in 2016. These chronic diseases that are due to unhealthy food intake include diabetes, obesity, hypertension, and cancer. Many are situated in vulnerable positions that are prone to health disparities due to political and governmentally driven structural factors. However, many Native communities affected have called for a resistance. Native Americans are today trying to combat health issues by reclaiming their food systems through years of colonization, misguided interventions, and globalized food systems, and in doing so, they are reconnecting to their own cultural identity through revitalizing and restoring traditional foods in ways I will get to later. Before I dive into the current food sovereignty movement, I will first go over some of the structural factors which allow us to understand how it is that people end up in generations of inequalities of health, why cultural revitalization through reviving and continuing the practice of traditional foods is unique and very much important to natives, and how food can be a powerful way to control the lives and well-being of a people. The first structural factor that I looked at was the settler colonialism phenomenon. Settler colonialism is a form of colonialism that seeks to structurally replace the original population of the colonized territory with a new society of settlers. The logic is that with this type of colonialism, it destroys in order to replace, and that primary motivation for elimination is territory. For years, the foods which were available to them were lacking, as the people were constricted to defined areas unsuitable for agriculture, as they also lacked resources to control their practices. The policies that created American Indian reservations resulted in drastic lifestyle changes that had a negative impact on health for Native people that has affected them back in the day as well as today through different forms. The second structural factor I looked at was government distributed commodity foods. The U.S. Department of Agriculture Food Distribution Program has been giving foods to low-income families on reservations and nearby rural areas since 1977. Referred to by many natives as commodity foods, these foods are no more than unhealthy canned food items and has continued to make up much of the modern native diet. Traditional foods become highly requested as natives claim that the food provided by the program was not necessarily representative of the foods their specific tribe ate. After the program began in the 1970s, people started to see a high consumption of these packaged and shelf-stable canned foods, excluding anything fresh, including no fresh vegetables, foods high in sodium, and instead more fat and sugar. A citizen of the Choctaw Nation in Oklahoma states that if you talk to people like me who grew up solely on this stuff, you hear stories of, I've never even tasted a pineapple or real spinach. You didn't taste these foods until you were older. And that we would scrape together whatever commodities we had available to us. The last structural factor I looked at was food deserts in both urban and rural Native communities. Before the 1940s and 50s, diabetes was rare in Native populations. Food deserts are areas that lack access to affordable and nutritional foods. In urban areas, corner stores are often resemble the food available in gas stations and are really stores filled with unhealthy snacks, including junk food and other sugary drinks. Diabetes is in large part a disease of acculturation, as Native peoples have become accustomed to an industrial Western lifestyle in which people are sedentary and rely on machines and store-bought foods. This high concentration of power in the hands of smaller amount of large-scale food systems leaves local food systems left with less power and ability to produce foods for the community. This leads me to talk about the food sovereignty movement and how Native communities today have based their traditional values of food with this movement. The term food sovereignty was coined in 1996 by a transnational organization of peasants, La Via Campesina, an international coalition that devoted particular attention to the struggles and rights of third world peasant farmers. The food sovereignty movement was initiated and became central to self-determination and decolonial mobilization embodied by indigenous peoples throughout the world. At the Nyalani Forum in 2007, those leading the movement presented the creation of 6 pillars. These are the basis of what food sovereignty strives for. The first pillar states that it focuses on food for people. Second, that it values food providers. Third, that it localizes food systems. Fourth, that it puts control locally. Fifth, that it builds knowledge and skills. Sixth, that it works with nature as it seeks to heal the planet so that the planet may heal the people. The case of this Native American health dilemma exemplifies the evolving food sovereignty movement. Indigenous people globally are actively shaping, nurturing, and fostering healthy and sustainable communities through their self-determination efforts and decolonialization strategies. More recently, the Native communities affected by this have called for resistance. Different ways that these indigenous groups have done so include creating programs such as community gardens, Native value and culture-centered education programs, joint health programs, a rise in indigenous chefs, and a rise in indigenous literature and academia that aim to share the importance and knowledge of traditional foods used that have been cooked and eaten by families prior to U.S. intervention. One community of natives from the University of Minnesota Morris have found that service learning empowers students to create a community of learning and practice. The students learned food practices important to Native culture, including Three Sisters. And the Three Sisters is an important cultural complex as the sisters are protagonists of a number of Seneca tales, myths, ceremonies, and legends, which include a garden of corn, beans, and squash. An important theme which the students learn is the theme of food as medicine, as the students learn of the significant connection between food and health for Native Americans. The former Indian American boarding school, now a university campus, has created this opportunity that is based on Native values, as the garden serves the community's needs for healthy, locally produced foods, as it engages students in understanding the development of the industrial food system through the service learning component, which places special emphasis on Native American history, horticultural practices, and cultural traditions. So how can anthropology better address food injustices? A 2014 report done by the DNA People adds that research that is intended to benefit the people should include community-based participatory research, since much of the existing academic literature on the people does not include their perspectives and ideas which isolate them from being able to make these decisions on major policies and programs that could impact their life. Many times, these policies and programs that intervene do not address the issues that were aimed to resolve and may even exacerbate the issues, as policymakers might be ill-informed. Finding a way that does not impede on the people's culture and lifestyle is not easy, as several issues can arise. Native scholar Eve Tuck states that communities that are being studied can exercise a bounty of decision-making power in these issues. And while most international efforts have intended to combat food insecurity through quantitative data, Singer and Erickson state that anthropologists advocate for additional grounded community-centered approaches to gain a more accurate understanding of specified local attitudes and experiences, considering there is a lack of understanding on the extent of food insecurity by policymakers, which brings effects on the lived experiences of those suffering from food shortages. White states that those who are part of the group that are vulnerable to such food injustices should have opportunities to voice their concerns to policymakers, hold more leadership roles in advisory and government, as well as have scientific resources to support the advocating of their rights. So finally, to conclude, the community's existing efforts of food revitalization must be studied more by anthropologists within the community being studied, so that Native voices on the issue of food injustices can better be addressed by policymakers. I would like to thank my advisor, Dr. Hannah Garth, as well as Dr. Katerina Semendeferi, and the honors program members, and of course, the Department of Anthropology and UCSD. Yes.
Given how long the period of time that the shift to these kinds of commodities has taken place, where is the knowledge being held, and how is it being, I mean, I can see how it's being mobilized, but what kept it alive over that length of time? You know?
For commodity foods?
No, for food sovereignty.
Food sovereignty.
Knowing what to eat, what the right thing is, for being able to say that this isn't good food and this is what we used to eat and that's much better for your health.
Yeah, so traditional foods or first foods have, they've been around since native, for years before U.S. intervention of, or Western diet kind of took over the their traditional foods. So it's not necessarily a new phenomenon, but it's mostly just that more people have called to this resistance more recently since the food sovereignty movement in 1996, which was started by the Via Campesina. So yeah, it's not necessarily like a new thing, but it's become more on the rise. Yeah.
I think the question was also who kept these traditions going, who has the information that is passed on, if I understand the question.
Okay, thank you. Yeah, so a lot of the ethnographic works I looked at, they center on community-based knowledge. So it was really mainly their elders who knew this knowledge and passed it down generations to generation. So, yeah. Any more questions? Yeah.
I have a question with regards to the contrast between farming people and non-farming people, given that Native Americans included both groups. And in foragers, there is really zero knowledge about farming, and foraging is not an option these days, except maybe in the high Arctic where you hunt. So how does the food sovereignty movement kind of try to mediate what, let's say, Native American groups here in California, where there was no traditional farming, there was very, very sophisticated hunter-gathering?
Yeah.
What would be their traditional food production, given that?
Yeah. So there are different tribes. It depends on what region they come from. So a lot of it is hunting, like back in in South Dakota, they do a lot of hunting, but also in desert regions like the Navajo, they've found ways to try to create these gardens to cultivate foods and grow. So it's mostly a lot of agricultural foods these days, but yeah, there has been issues with trying to create or trying to forage for foods like through hunting, yeah. So hope that answers a little bit. Any more questions?
Wonderful job, everyone. I'm so proud of you. So proud of you. All right. So this is going to be the second and last part of our gathering today. I would like to, on behalf of the department, acknowledge some of our exceptional undergraduate students. So I'm going to go right ahead with that. And Garrett, I'm going to need your help, as you know. All right. So one of the awards that the departments give to exceptional students in biological anthropology, well, in anthropology is the Washburn Award for Excellence in Biological Anthropology. And you have a list here of students who got the award throughout the years, since '96 all the way to 2017. And this year's award goes to Jennifer Corona. I think that Jennifer is still taking an exam in a class, so I'm going to hold this on the side, and when she comes, I'm going to give it to her. All right. Our other award is the Robert McAdams Award for Excellence in Anthropological Archaeology. And this year, the award goes to Matthew Van Schalk and Christina Fallone. Matthew, there you are. Congrats.
Oh, thank you.
Congrats, Christina.
Thank you.
The Hallowell Award for Excellence in Sociocultural Anthropology this year goes to Rachel Oporto. Again, I'm going to hold on to the award, hoping that both of the students will be able to make it. I know it's a time where classes are going on and other things. So, and this year, our department is introducing a fourth award in honor of the founder of our department, Mel Spiro. So the award is to recognize excellence in psychological and medical anthropology. And this award goes to Joshua Beneventi. We had several exceptional students in the honors program this year, and we would like to congratulate all of you and really give you our best wishes for the future, and just a little symbolic item and a letter of recognition for what you did. So I would really appreciate it if all of you come in the order that you are listed up there: Cesar, Josh, Jennifer, Barbara, Christina, Amy, Emily, Matthew, and receive the cord and the letter of recognition. All right. So, Cesar, this is for you, and this is the honors cord. And we can give you a hug for a great job. Thank you. There you are, Josh. Congrats again. One more. There you are, Barbara. Congratulations.
Thank you.
Thank you. Hello, Christina. Congratulations.
Thank you.
Thank you. Congratulations, Amy. Thank you, Karen. Congratulations, Emily. Congratulations, Matthew. All right. We have another special award, the Lisa Stefanacci Award for Excellence in Human Comparative Neuroanatomy. And we have had several students since 2014 who took the award. I would like to ask last year's awardee, Mr. Dion Cuevas, to come here in the front and present the award to this year's student, Miss Demi Greiner. So if Demi can step up here. And I want to hug too.
Thank you.
Thank you, Dion. Okay, we need to recognize our ambassadors of anthropology. So those of our students in the honors program who go out of their way to represent our department with outreach events in Triton Day and lectures that are being given across campus in support of our program at Anthropology. We have pictures from students from previous years, and I would like to just bring up Cesar, Josh, Barbara, Amy, Emily, and Matthew, and a special thanks to you guys, and congratulations for being in this program and helping us out. Thank you. Lots of thanks and acknowledgements go to the Anthropology Club, and these are the officers for 2017-18. Tanya, Cesar, Jessica, and Amy, and all of the other students who participate in the program. We thank you, and we appreciate all you're doing for the Anthro Honors Club. So. I would like to mention really quickly, I'm not going to go through all the names, but really the important contributions to the undergraduate program and education anthropology. There's a lot of people involved, and I would like to first of all bring up our lecturers who are offering year after year all these courses, and they are such a critical component of how we train our students in the department. So thank you so much, you guys. All of those people are listed there, the ones who taught for us this academic year. Thank you very much. We really appreciate you. Our graduate students, they are amazing. They are hardworking, and they have a lot of work on their own, and they also serve as TAs and teaching assistants. And we really rely heavily on you guys to help us educate our undergrads. So thank you so much to all of those who served as TAs this year. The Mentor Protégé program is a program that was initiated by the graduate students in this department. They mentor undergrads. They established a whole set of ways to recruit and support undergraduates. We're grateful to them, and the director, Dylan, the program coordinator this year is Belinda, and all of the students participating in the program. Thank you. Thank you very much. Our alumni. I see several of you in this room. We are so excited that you're staying in touch with us. Thank you for the support. We have our last year's class from 2017. Several of the students contacted me. They are sending us our greetings. Some of you are in this room, and I look forward to catching up with you in the reception and introduce you to students. I would also like to go over a couple of video clips that a couple of our alumni sent to us with their greetings and salutes. And I'm going to try it again.
I'm a member of the 2017 Honors Thesis cohort. After I graduated UC San Diego, I started a Master's of Arts in Biology program with the Advanced Inquiry program. This program partners with San Diego Zoo Global and Miami University to offer a really innovative degree in biology, but it's focused on conservation. I remember being in your shoes a year ago, being very nervous, but also excited to wrap up my undergraduate degree. I was unable to make it to the event today, but I still wanted to extend my sincerest congratulations to all of you for finishing the thesis program. It's a lot to take on during your senior year, but you've made it this far, and you're very close to the finish line, only a few weeks to go. Hang in, you can do it. Congratulations on how hard you've worked these past few quarters and years. Your accomplishments are really going to pay off. For me, the undergraduate thesis program was one of the best things I could have done to prepare for grad school and even for jobs. Thank you to Katerina and to all of our wonderful department staff and to our faculty mentors for continuing to support this really important program. We couldn't do it without you. And to you, 2018 cohort, enjoy your couple weeks before you have graduation, and congratulations. Bye.
And there is one more.
First things first, congratulations to this year's Anthropology Honors Cohort for 2017-2018. You guys worked so hard to be here, and I'm really proud of you guys. I bet the results were phenomenal. You know, I'm sorry I couldn't be there because I'm in the distant land of Los Angeles. Well, actually, no, no, I could be there. I'm like right there. Yay, I'm right there. Since last year, I've just been wandering around and I found myself working at the Museum of Latin American Arts in Long Beach. And you can ask me more about it if you care, once you guys are done. But this isn't the end of my journey, and this is definitely not the end of yours. So just keep at it, and I'll see you at the end of the presentation.
All right. I have a couple of more slides. None of this could have happened without our wonderful undergraduate advisors, Garrett Soriano and Brittany Wright. So thank you so much. Mr. Garrett Soriano is a new addition to our department, so I would like to take the opportunity to welcome him in our department. He came to us after working as a counselor, and he has a bachelor's degree in sociology and a master's degree in counseling, so we're very, very happy to have you, Garrett. Thanks to all the faculty. We had several mentors, obviously, who work with the students. And I would like also to extend our thanks to our staff in the department. You are more important than we can ever express to you. So thank you so much for your help with everything we do here. For the staff. Okay, and at this point, I would like to invite everyone next door in room SSB 105 to join us for a reception and celebrate the achievement of our students. Thank you very much, everyone.